All right, hello, BIO205, and welcome to your second lecture. So today we are going to be discussing in greater detail the ways in which we study ecology. But before we jump into course content, I did want to go over a few housekeeping details with you. So firstly, today Practical 1 is released. So this activity will be due on Friday, September 18th at 8 p.m. So please do be mindful of the fact that the assignment is due at 8 p.m. Depending upon the other courses that you're taking, you might be more familiar with assignments being due at midnight or 11:59 p.m. In our course and for all assignments, we will have everything due at 8 o'clock. And this was actually a recommendation that has been made by the university in order to help encourage better sleep schedules in our students, because we don't want you to be rushing or pressure to finish your work, staying up until midnight to get things submitted. So hopefully this 8 p.m. timeline will help to incentivize you to complete that work a bit earlier and then have a few more hours in the evening to decompress and relax before heading to bed. So all of our deadlines in BIO 205 will be for 8 p.m., but the deadlines will vary. So this one will be released today on Monday and be due on Friday, September 18th, and will involve watching some videos of habitats across our campus and doing some research on the types of organisms and abiotic conditions that you would expect to see in 4 different habitat types around our campus. And so this is to replace the usual labs in our course in which we would have all gone out together and visited these locations. But of course, with distance learning, we can't gather in large groups to go check out these locations. So we have instead created videos and other resources for you to learn about these same habitats. Since Practical 1 is being released this week, with that, your TA help sessions will also begin this week. So if you have any questions or concerns regarding your assignment, you can join in on the practical help session in which you are officially enrolled. These are optional, so attendance will not be taken. Or, of course, you can always email your TA on another day of the week. So if a question comes up for you, either before or after your help session, you can, of course, email them at any point in time, being mindful of the deadline. So you don't want to email them right before the deadline of the worksheet, as you might not get a response. As well, starting today are your lecture participation quizzes that will count towards that participation grade. So all of these participation quizzes will only be available for the 24-hour window. We'll release them at midnight and they will be gone at the end of the day. So as soon as it becomes Tuesday, that quiz will be locked and you will no longer be able to access it. As mentioned previously, the accuracy of your answers doesn't matter, so you don't have to feel stressed about getting the questions correct. This is an opportunity for you to test your understanding of the lecture material, so it can be a great way to check to see if you're keeping up and understanding the material. And if you are struggling with some of those questions, you might consider rewatching portions of the lecture or visiting your textbook for some additional help. And unfortunately, late quizzes cannot be accepted. So once the clock strikes midnight and it is now Tuesday, that quiz is locked and it cannot be reopened. So the purpose of these quizzes is to not only act as a check of your understanding, but also to incentivize students to be signing in and viewing their lectures every Monday and Wednesday to ensure you don't fall behind on that content. So that's why they're only going to be available for that short period of time. And now, since this is the first week in which you have assessments to complete, materials that are going to be graded, I do want to again remind you to check out our course policies on late and missed assessments to ensure that you know what is expected and what you need to do in an instance of, say, you become ill or there's a family emergency. And in order to figure out what our course policies are, it's quite simple.
A message from Big Snoop Deal Double G to all the university students. Yo, syllabus. You gotta do it. You gotta read it. Man, I'm telling you, the more you know, the further you go. Tell them Snoop Dogg sent you. Syllabus. I know all about it. Catch up so we can have a conversation. Don't be late. Let's go.
Couldn't have said it better myself, thanks Snoop. So make sure that you have read our course syllabus if you haven't already, so you are familiar with all of our course policies.
Message.
All right, so last class we talked about the scale of ecology. And we looked at all of the different levels at which an ecologist could investigate a system, from the individual all the way up to the biosphere level, in which there will be increasing complexity. So although there are all these different levels at which you can study ecology, and they are quite different from one another, there are still three unifying principles that are applicable at all three of these, or all of these spatial scales, I should say. And these three principles are, firstly, the conservation of matter and energy, dynamic steady states, and evolution. So let's break down each one of these principles in a bit more detail. All right, so the law of conservation of matter, as well as the first law of thermodynamics, they're very similar. It's the idea that for the law of thermodynamics, that energy cannot be created or destroyed, only converted to different forms. And it's the same with the conservation of matter, that matter cannot be created or destroyed. And this is applicable to ecologists in many ways. For example, if you were studying the energy transfer through trophic levels. And this is a concept that we're going to touch upon more in later lectures. But what you often see if you build your ecological interactions with all of your trophic levels, beginning with your primary producers to your primary consumers, secondary consumers, tertiary consumers, and decomposers, is that it seems as though there is a loss of energy from the primary producers up to higher trophic levels. And it's often described that way as well, is that there's an inefficiency and there's a loss of energy from subsequently higher trophic levels. But is that really an accurate way to state it? Is energy actually lost from this ecosystem? Well, no, because as we know from the first law of thermodynamics, that energy cannot be lost. It's not lost. You could, if you wanted to, continue to track that energy throughout the community, the ecosystem, the biome, whatever level of ecology in which you are studying. So it's not being lost, it's just being converted into different forms. For instance, a major source of this apparent decline in energy between trophic levels is the fact that a lot of this energy is converted into a different form, for instance, in the form of heat or through the process of growth or respiration. All right, so the second unifying principle is that of the dynamic steady state. So as was kind of alluded to in the last slide, there's this continuous exchange of energy and matter throughout an ecological system in all sorts of varying forms or as it's converted from one form to another. But there's also a balance of gains and losses within an ecological system. For example, say we wanted to even look at the level of an individual, if that's our unit of interest in what we're studying in ecology, and we're interested in the individual red squirrel pictured here. This guy looks a little bit cold, all covered in snow here. So if we want to look at this red squirrel, we might say that this little guy is experiencing heat loss to the environment. However, this individual is also gaining heat through their metabolic processes. And so in that way, yes, they're losing heat to the environment, but they're also gaining heat. And so they can maintain an internal body temperature within a relatively narrow range. And so there's this continuous exchange of energy here, this continuous loss of energy via heat to the environment, but also continually gaining heat through their metabolic processes to result in that dynamic steady state. So constant exchange of energy, but overall maintaining a relatively stable level, in this case, in this example, in terms of their internal body temperature. You could say this for many other factors. For instance, they will lose matter, or they will lose food that they have intaken via their waste. So they're going to release waste, but that's always being replenished by the new food that they consume. So that's an example of an exchange of matter within this individual squirrel as well. But again, it's always in balance or in equilibrium, representing this dynamic steady state. And it doesn't just apply to the level of the individual. This will apply to all levels of ecological organization. You can always find examples of this balance, this dynamic steady state, where there's this constant flux in energy and matter, but at the end of the day, it always balances out. So again, with the individual, if the input is food, their output is waste, like I just mentioned in the last slide. If you're looking at the level of the population, perhaps you might quantify how births and immigration balances death and emigration. At the level of the community, one example of this could be colonization by new species is balanced by the extinction of existing species. At the level of the ecosystem, that dynamic steady state might be represented by solar energy and matter entering the ecosystem, balancing any energy and matter that might be leaving the ecosystem for other neighboring ecosystems. And then at the largest spatial scale, the biosphere, our planet will also represent a dynamic steady state, as for instance, the solar energy entering our planet is balanced by the energy that is re-radiated into space. All right, and our third and final unifying principle across all these levels of ecological organization is evolution. Now, as you have learned when you completed your first year biology courses, evolution is a result of natural selection, as well as several random processes that are at play here as well. So, of course, natural selection is the mechanism of evolution, but other important processes to consider includes the process of mutations. Mutations are incredibly important because at the end of the day, they are the ultimate source of all new genetic variation because they're providing that raw material for which natural selection can act upon and are responsible for sometimes or often neutral mutations, often detrimental mutations, but also those that result in the creation of a new and beneficial trait in an organism. There's also genetic drift. So this is the change in the frequency of an allele in a given population due to random sampling. So the alleles in the offspring of any given population are just a sample of those in their parents. And so there is that role of chance in determining which alleles the offspring will acquire from their parents and will be passed on. And because of this random chance involved in the alleles that the offspring receives from their parents, you'll always have fluctuations in the frequency of a given allele and how much it's represented in a given population. So just due to random chance, you can see changes in the frequency of a given allele in a population. Population can also experience a bottleneck. Now I write sort of here because it's sort of a random process. So there's definitely a random nature to it, but there can be aspects of a bottleneck that are non-random as well. So firstly, a bottleneck effect occurs when there is a drastic decline in the size of a population. And this could be caused by a natural disaster, drought, disease, or maybe human activity like habitat destruction that wipes out a large proportion of that population. As a result of this bottleneck, it reduces the diversity of the gene pool. And so the representation of the alleles that make it through this event, that make it through that bottleneck, so really the individuals that survive this event and the alleles that they carry, it could be random, but it could also be non-random. So again, that's why I said it's sort of a non-random process. So for example, if all of the survivors of the bottleneck are individuals that had greater genetic fitness for some reason, dependent upon what alleles they carried, then you could have a non-random aspect to this bottleneck event. But overall, the genetic drift that's triggered by the bottleneck can change the proportion of the distribution of the alleles, and it can even result in loss of alleles entirely. And lastly, the founder effect. Again, sort of, there's definitely a random component to it, but there can also be a non-random aspect. So the founder effect is when you see a loss of genetic variation in a new population that has just established itself from a few individuals that have originated from a larger population. They've moved over to a new area. They've established a new population. But because there's so few individuals, there's overall a lower level of genetic variation compared to the original larger population. So in some cases, the founder effect can actually lead to speciation, which I'm going to talk about at the end of this lecture. But that founding population, that new population that is moved to a new location, generally represents a random sample of the original population. So except the fact that you'll typically have lower genetic variation represented. However, there could be that non-random aspect regarding a few traits. For example, if individuals in this population had traits that assisted in their ability to establish this new population, perhaps it's only individuals that had a high dispersal ability. So maybe certain traits might be overrepresented in this new population. But other than that, the remaining alleles will represent a random selection from that original population. So these are occurrences that we see, that one of these unifying principles: evolution as a result of natural selection, but mutation, genetic drift, bottleneck effect, and founder effect can all be playing a role in the level of genetic variation that we see and the overall frequency or representation of certain alleles in a given population. All right, so there's also different types of selection. There are three main types: disruptive, stabilizing, and directional. So let's begin with disruptive, the first one here. So when a population is experiencing disruptive selection, this occurs when the extreme values of a given trait provide the greatest advantage compared to the intermediate values of a trait. So in this simple figure here on the right, the red line represents a founding population, or the start of the population, that has kind of a normal distribution in terms of a given trait. And then over time, that distribution of that trait value is represented by the blue line, in which the two extremes of that trait become more and more commonly represented, and the intermediate level of that trait diminishes because it provides the least advantage to these individuals. So over time, the population can divide itself into two groups that look quite distinct from one another. Now, overall, in nature, it's actually quite rare to find examples of disruptive selection. But it does happen occasionally, and when it does, it can actually lead to sympatric speciation. So sympatric speciation is when you see what was a population of a single species over time separating into two different species, even though they were not geographically isolated from one another. They were living right next to each other. They could interact with one another if they wanted to. But over time, despite the fact that they're not geographically isolated, they still become two separate species. And what can cause that is if disruptive selection is at play, when two extremes of a certain trait are favored, resulting in a population that has two groups that are increasingly distinct from one another. So to try to visualize this, it always helps me to have a kind of concrete example. So what is a scenario in which disruptive selection occurs? Well, we actually see this form of selection with oysters that are found in shallow waters or intertidal zones. So in these oyster populations, the intermediate color, so the kind of medium brown color, is actually quite rare to see. And what you find more often are either very light colored oysters or very dark colored oysters. And this is because those that are of an intermediate brown color are more visible to predators like seabirds. So the light colored oysters can blend in pretty well into the rocks that are found in these shallow water areas. And the very dark colored oysters can blend in better into the shadows that are cast by these rocks. So both of these extremes of this phenotype have areas in their habitat in which they can be camouflaged. But the intermediate range of these colors, they show up against either backdrop. And for that reason, that makes them easier prey. So with them being easier prey, they're picked off, their numbers dwindle, and with fewer of these intermediate brown colored individuals actually surviving to reproduce, over time, we see a population that has more oysters colored at either extreme of the spectrum and more rarely seeing individuals of this medium brown color. All right, so let's move right along to the second form of selection, which is stabilizing. Now, stabilizing selection is much more common than disruptive. And this occurs when the mean value of a given trait that is seen in a population stabilizes on that medium or intermediate value. An example of this is clutch size. So clutch size is the average number of eggs that is laid by a female bird. And in all bird species, we see this mean value averaging out to, say, a medium number of eggs. And the reason for this is that you don't want to lay too few eggs because the goal is to have high reproductive output to pass on your genetics to the next generation. And it's also best to have several offspring as opposed to, say, just laying one egg in case that offspring doesn't survive. However, it is also not beneficial for a bird to lay many eggs because then it becomes much more difficult for her to care for those offspring, and it's very unlikely that they will all survive. So in birds, they exhibit a lot of parental care. If you've ever seen, say, a robin's nest being built near your home, you would have seen that the parent bird flying off, collecting food for their young and bringing it back. So they take care of their young. And if there are too many young chicks to feed, they won't be able to provide for them all, and therefore they will not all survive. So because having too few eggs really doesn't assist in maximizing reproductive output and spreading your genetics to the next generation. But then again, also having too many eggs becomes much too difficult for you to care for all of your offspring. For this reason, over time, the average number of eggs laid by a given bird species falls somewhere in the middle, not too few, but not too many. And this is an example of stabilizing selection. All right, and then the final form of selection is directional. And this occurs when one extreme of the phenotype is favored over the others. So you have this continual shift, generation after generation, towards that extreme end of the trait. An example of directional selection can be seen in a classic example of evolution that you may have been taught in a previous class, perhaps in first year biology, maybe even in high school, and that is of the London peppered moths. So the London peppered moth evolution was triggered by the Industrial Revolution in London, which occurred around 1760 to 1840. And during the Industrial Revolution, soot started to cover all of the tree trunks throughout the city. The result of this is that it killed off all the lichen that used to grow on the bark of the trees, and this lichen had caused the tree trunks to have that light gray color that was also speckled with a bit of black, as you'll see here on the left-hand image. And instead, everything was just coated in black. And for that reason, what we saw in the industrial populations of the peppered moth, or those that lived within the city, is that if you were a light gray pepper pattern moth, like the one we see here, you used to be able to blend in quite well into a lichen covered bark, but now you stood out as a very easy target against a dark soot colored tree for any birds or other hungry predators. However, any of the moths that had darker pigmentation gained a competitive advantage as they were better able to blend into all the soot-covered surfaces as well as a lot of the concrete that was being laid within the city. So over time, what we saw, although the rural populations would remain with this speckled pattern, what we saw in the industrial populations of these London pepper moths is a preferential selection for darker and darker coloring. So there's this directional shift towards greater and greater black pigmentation in these moths in order to allow them to best blend in or camouflage to their environment of soot and concrete. All right, and just to drive that point home, we'll go through one more example of each of these three types of selection, using the example of tails for all of them. So we'll begin with directional selection. So an example of directional selection for tails would be, say you have this lizard species. And for this lizard, it is of greater competitive advantage to have a long tail because the longer the tail is, the more that they are able to manipulate it or wiggle it around to look like a snake and scare off predators. So the longer the tail, the more it looks like a snake, and the greater likelihood of survival this hypothetical lizard has. So this would be an example of directional selection, in which generation after generation, you would see the selection for increasingly longer tails in this hypothetical lizard. But say, for example, you have a large cat species, and in this case, it's no longer beneficial to just have a longer and longer and longer tail. Because in this case, having an extremely long tail means that it's going to drag on the ground and slow down the ability for this cat to run. However, on the other end of that, having a tail that is too short will disrupt the balance of this cat. They still do need their tail to help them balance, but they don't want it to be too long that it ends up slowing them down. So this would result in an example of stabilizing selection, in which the greatest advantage is provided for those individuals that have a medium length tail. And then a final example would be for disruptive selection. So again, disruptive is when the extremes of a given trait are selected for and the intermediate levels are selected against. So say we have a squirrel species, and those that have a very short tail experience a competitive advantage because it makes it easier for them to escape from predators. So in this scenario, predators often snatch up these squirrels by grabbing onto their long tails. Well, if you don't have a long tail to grab, you're more likely to escape that predator that's trying to get you. But on the other extreme, having a very long tail will also be beneficial because it will help for balance as these small rodents move around in the trees. So in this case, there will be selection for very short tails as well as for very long tails, but those individuals with a medium length tail really don't experience any advantage. It doesn't help them run through the trees in their habitat, and it also doesn't help them escape from their predators. So a medium length tail will be selected against, and either short or long tails will be selected for. So those are just 3 more examples of these types of selection using the example of tails. All right, so in terms of the speed in which evolution occurs, this was a concept that for a very long time, it was thought that evolution took lifetimes, several lifetimes, in order for change to be seen. But that concept was changed thanks to the work of Peter and Rosemary Grant pictured here. And they have spent their entire career studying Darwin's finches in the Galapagos Islands. And through their research, they show that evolution can occur much faster than previously believed. So for their research, they went and visited the Galapagos Islands, and they measured the length of the beaks for ground finches. And so this is a figure of their data here. So on the x-axis, we have beak depth in millimeters, and on the y-axis, the frequency of that beak depth. And so we can see in the first year of their data collection, the mean beak length was about 9.5 millimeters. But then there was an extreme environmental change in which there was a severe drought on the islands that lasted for a very long time. So these ground finches, in which they are measuring the beaks, they are seed feeders. And in the first year that the Grants visited the island and conducted those measurements, there was an average of at least 10 grams of seeds lying on the ground for every square meter. So food resources were highly abundant at this time. Then the drought hit, and when they revisited in year 2, we were down to on average 6 grams of seeds per square meter of land, and by the third year, only 3 grams of seed. So you can see this is going to be problematic for our ground finches. However, not only were they experiencing a reduction in available resources, but they were also experiencing a shift in the types of seeds that were available. So not only were the seeds decreasing from an average availability of at least 10 grams per square meter all the way down to 3 grams per square meter, but within those 3 grams, there is now a change in the composition of those seeds so that the majority of the seeds available belonged to this plant here called caltrop. So the problem with caltrop seeds, pictured here on the right, is that they are incredibly difficult to crack open. And in fact, it takes a bird with a beak of at least 11 millimeters long to even have the strength or the ability to crack open the seeds of this plant. Now, the reason why the caltrop plant was overrepresented at this time is that this is a plant that is highly drought tolerant. So while this drought extended year after year, many other species started to die off. So that's why we saw the reduction in seed availability, but we also saw this shift to mostly caltrop seeds available because this plant will do just fine under drought conditions. So in the Grant's observations during this drought, they found that ground finches with beaks less than 10.5 millimeters long were not even found to be trying to crack open and eat the caltrop seeds. So after this drought for 3 years, the Grants went back in year 3 and remeasured the distribution of the beak length. And this is what they found. So previously, the average beak depth was 9.5 millimeters, and after the drought conditions, the average beak length increased to 9.9 millimeters. You can also see an overall shift in the data as well to those individuals with larger beaks. And this is due to the fact that, not surprisingly, since there was a shift in the availability of seed resources, only only those birds with larger beaks were able to take advantage of that resource that was now available, the caltrop seeds. And so they were able to survive this drought. But many individuals that had smaller beaks and were not able to consume these seeds were not able to survive and therefore reproduce. So the grant saw this shift in the average beak length. So there's a change in the frequency of this trait, and beak length is a heritable trait. So this was not a plastic change. This was a heritable trait. So what the grants found is that evolution can actually occur quite quickly, and was observed in only a few seasons, whereas previous to this, Darwin thought that it took lifetimes for any evolutionary change to occur. What they saw was that even within just a 3-year timeline, there was an evolutionary shift seen in this population of ground finches, as well as the fact that evolution can occur at smaller scales. So much of what Darwin spoke of was the evolution of new species. But of course, evolution doesn't just occur at the level of the species. We see evolutionary change occurring all the time within a given population. So that's the other idea that was promoted by the Grant's discovery. So this idea of evolution occurring at a smaller scale all falls into the umbrella of what we call microevolution. So microevolution is really just evolutionary change that occurs in populations. And it can occur via natural selection or artificial selection. So artificial selection being those changes that are influenced by human intervention. So some examples of this. So if we think about artificial selection, so changes that humans have made to a population of a given species, what comes to mind? Well, one example is in our plants that we use as crops. So a lot of our crop species don't look anything at all to what their original or wild type plant looked like due to this artificial selection, the selective pressures implemented by humans in order to increase the frequency of desirable traits. And an example of this is in brassica. So brassica, it's a wild mustard species, and through artificial selection, we have actually resulted in this single plant species being representative of several types of produce that you are likely familiar with. So Brussels sprouts, cauliflower, broccoli, kale, cabbage, and kohlrabi are all from the exact same species of plant. The only difference here is that the populations have been selected for different traits. For instance, to create kohlrabi, you would simply select for individuals that have very large stems. For cauliflower, you select for populations that have the stunted floral growth. And for kale, you select for populations that produce very large leaves. So all of these different types of produce are all coming from the exact same species. It's simply human intervention via artificial selection to select for desirable traits. There are also examples in which humans have unintentionally triggered artificial selection in populations. And an example of this is through antibiotic resistance. Now, this is a concept that you might be familiar with already, as it is of increasing concern in the medical community. So what is antibiotic resistance? Well, in a nutshell, it's the fact that over time, more and more of the antibiotics in which we have created to fight common infections are no longer effective against the bacteria. The bacteria are now resistant to the medications that used to work for them or against them, but now are no longer working. So why does this occur? Well, say for example, you get sick. And that is because your body has now been infected with this population of bacteria. But bacteria replicate very, very quickly, many generations in a very short amount of time. And because they replicate so quickly, via genetic mutations, there's always going to be a large source of genetic variation in these populations. So say, for instance, you're sick with this population of bacteria, but there are a couple of individuals who now have a genetic mutation that has caused them to become more resistant to the antibiotic than any of their fellow bacteria. So you're infected, you're not feeling well, you go to the doctor's office, and they prescribe antibiotics that typically work for this type of bacterial infection. You start to take your antibiotics and you start to feel a lot better. And that's because these drugs have successfully killed off the majority of the bacteria that were found in your body, to the point where there's only a few individuals remaining. So, firstly, as kind of an aside, it is always incredibly important to take all of your prescription. If you ever get sick and a doctor prescribes you antibiotics, make sure you finish every last pill in that bottle, even if you start to feel better partway through the prescription, because you want to ensure that you kill off all or almost all of the bacteria that was causing the original illness. Because say, for instance, you take some antibiotics, you're feeling better, you've killed off most of those bacteria, but there are still a few individuals hanging around. So at this point, you're feeling better, but these few individuals are still there. And as you can see, the two individuals that survived or that made it to the end are our two resistant bacteria. So you stop taking your medication, and then those bacteria begin to replicate. But now the majority of the individuals are those that possess that genetic mutation to cause them to be resistant to that antibiotic. So that population is growing and growing inside of your body, and you start to feel sick again. So you head back to the doctor's office and they say, oh, it looks like, your illness came back. Here's another round of that antibiotic. You start taking that drug, but now it doesn't work for you anymore because you are no longer infected with the same strain of bacteria that you were in the beginning. You are now infected with a strain of bacteria that is resistant to this type of antibiotic. So this is a process that we're seeing occurring more and more frequently, and why several of the antibiotics that we've been using for quite some time now are experiencing a reduced efficacy against the bacteria in which they previously were able to kill with little issue. All right, so those were a couple of examples of artificial selection, but of course we can see microevolution occurring in populations via natural selection. For example, through the selective pressure that a predator exerts on its prey population. So say, for instance, you have a population of amphipods, and this is their predator over here, this larger fish. Larger fish prefer to eat larger amphipods just because they're getting more bang for their buck. They're getting a larger individual, more nutrient dense, so they're always picking off the larger individuals in a given population. And they don't really bother with the smaller amphipods. Well, over time, that selective pressure is going to result in a population of amphipods that are overall smaller in size. because all of the larger individuals are always being selectively removed from the population via the predator, leaving behind only those that are, on average, smaller in size to survive and then reproduce. So this would be an example of microevolution being caused by the selective pressures of a predator. All right, so that was a lot of information on microevolution, looking at the Grant's discovery of the ground finches, artificial selection, the previous example of natural selection. But on the other extreme of this is macroevolution. So when we think of macroevolution, really what we're talking about here is higher levels of taxonomic organization or otherwise speciation events. So when a new species is formed, this is what we would classify as macroevolution. So speciation can occur through one of two channels. The first being allopatric speciation, and this occurs when a new species evolves because geographic isolation has occurred in a given population. So say, for example, you have this population of field mice. They are all living together, interacting with one another, able to mate with one another. But then through, say, tectonic activity, the rift valley forms or a river starts to run through this region, and those mice are now no longer able to cross this geographical barrier. And so now they're separated from one another. This is the perfect recipe for, over time, speciation to occur, because there's now no longer any gene flow between these two separated populations. They are no longer able to mate with one another. So due to the fact that there is no longer gene flow occurring between these populations, and the fact that they are likely adapting to the local conditions of their given environment, over enough time, they can become two separate species. So that if you were to try to reunite those populations later down the line, individuals would no longer be able to successfully reproduce. On the other hand, you can have sympatric speciation, which I mentioned briefly in a previous slide. And this occurs when you have a population that separates into two or more species, but there's no geographic isolation. They're all still living in the same area and hypothetically still able to interact, and yet they separate into two or more distinct species. Well, how can this occur? There's a few mechanisms, but one example is through African kitchlids, pictured here. And this is a great example of sympatric speciation, in which you can have many species of this fish living together in a single body of water. And they have become different species due to sexual selection. So there is a very strong influence of sexual selection in this group of fish. And that is due to the fact that the females exert a sensory bias in that they will only mate with males that are of similar colors or markings to them. So in the images here, you'll only have, say, these black and red females seeking out and mating with other black and red males. And they will never mate with a male that is, say, blue or yellow in color. So because of this extremely strong mate selection, these fish can still be living within the same body of water, but they will never mate with one another. They'll never experience any gene flow because of that strong female sensory bias. And so over time, they have resulted in the creation of several distinct species of kitchlids found within a single lake. All right, and that is it for today. So our next class, we will talk about the evolution of adaptations. On your to-do list, as mentioned at the top of this lecture, is to begin working on practical assignment number 1, which is your preliminary habitat observations of your UTM campus. This will be due Friday at 8 o'clock p.m. Remember, if you have any questions on this, you can email your TA or drop into your optional Zoom session during your scheduled practical period. Also, of course, do not forget to complete your participation quiz. Accuracy of your answers does not matter, but it will only be available until the end of the day today, 11:59 p.m. And unfortunately, late quizzes cannot be accepted, so make sure you get that done before the quiz is locked. All right, that's it for today, and you will all be hearing from me again on Wednesday.